I'm Daniel Levine, and this is the Bio Report. Myeloid cells can stimulate anti-tumor effects in the body and recruit additional immune system cells to turn a cold tumor hot. Bolt Biotherapeutics is developing a new category of immunotherapies that combines the precision of antibody targeting with the strength of the innate and adaptive immune systems. By activating and recruiting myeloid cells, the company's experimental therapies are designed to reprogram the tumor microenvironment and invoke an anti-tumor immune response. We spoke to Randy Schatzman, CEO of Bolt Biotherapeutics, about the company's immune-stimulating antibody conjugates, the case for activating both the innate and adaptive immune systems to fight cancer, and the company's lead experimental therapy for HER2-positive cancers. Randy, thanks for joining us.
Danny, it's great to be speaking with you again. Appreciate you having us.
We're going to talk about Bolt Biotherapeutics, its platform technology, and its efforts to develop immunotherapies that activate both the innate and adaptive immune system. Perhaps you can begin by explaining for listeners the difference between these two different parts of the immune system.
Yeah, I think it's important to recognize the differences here. The innate immune system is I'll call it the first line of defense in the immune system. It's the one that each of us are born with and that we inherit from our parents. And it's really involved with recognizing foreign invaders, particularly microbes, et cetera. And it's a quick response system that eliminates those. Interestingly, components of the innate immune system are then able to, as they destroy the foreign invaders, present certain antigens from those foreign invaders to the adaptive side of the immune system. And that's I'll call it the more highly trained, focused part of the immune system that recognizes very specific antigens on these foreign invaders. For example, it's the part of the system that would recognize the spike protein on COVID-19 to help destroy it.
The early market successes around immunotherapies have been focused on activating the adaptive immune system. How successful have these therapies been?
Well, in fact, they've been very successful and have solved and eliminated cancers for just a huge number of patients. The challenge with them is that they only work with, I'll call it a subset of all cancer patients. And I think what we're working hard with in the field is to expand our treatments so that ultimately we can impact all cancer patients instead of just a subset.
Much of the success to date for these therapies have been targeting liquid tumors. Is there reason to believe part of the explanation for why we haven't seen the same type of success in solid tumors have to do with how the immune system is activated by these therapies?
Well, it's partly that, but it's partly that the solid tumors are a really complex environment. The architecture obviously is very different than dealing with a cancer cell that's circulating, in the blood or in the bone marrow. Every tumor is different in terms of its structure, the number of cancer cells that are there versus suppressed invading immune cells is all completely different from tumor to tumor. And so it's been a much more complicated space to try and impact.
Bolt's platform technology involves what it calls its Bolt Body Immune Stimulating Antibody Conjugate, or ISACs. Can you explain what these are and how they work?
Yeah, very simply put, Danny, these are antibodies to which we what we call conjugate a payload that activates the immune system. In this case, the antibody is really about targeting that payload to a specific tumor and an antigen on that tumor And then the payload itself, once brought into the tumor microenvironment, activates the immune system. And what we're doing is actually bringing the patient's own immune system to their tumor to recognize it and eliminate it by doing this.
We've seen antibody conjugates where the payload is a chemotherapeutic agent. My sense is that in doing these therapies, a lot of the challenge and magic, as it were, has to do in the linker. What does it take to link your payload to the antibody, and how predictable is the delivery and release of where you want them to go?
Yeah, it's an interesting question. So the space that you're speaking about is what we call the antibody drug conjugate space, where we've taken antibodies and put on what we call cytotoxic payloads. And in this sense, the antibody takes that cytotoxin, delivers it to the cancer cell itself. It binds the cancer cell, and the cancer cell then internalizes that whole structure. And ultimately, it's the linker is set in a way that the change of pH, the acidity within the tumor cell allows the linker to deconjugate and release the cytotoxin so that it kills the cell, but also is able to diffuse through the tumor and kill the surrounding tumor cells as well. Interestingly, with an ISAC, we had to unlearn many of these principles, because all we want the antibody to do is direct our payload to the cancer cell, but we don't want the cancer cell to internalize it. In fact, what happens is after it binds the cancer cell, we're looking for an interaction of the other end, what we call the FC portion of the antibody, to interact with surrounding cells, in this case myeloid cells, which once bound ultimately internalize what we call the ISAC into the myeloid cell. And it's there that the magic happens. Within the myeloid cell is a group of receptors that we call toll-like receptors that are able to stimulate the innate immune system. And the payload that we put on our antibodies is a direct activator of that. It's a small molecule, activates what we call toll-like receptors 7 and 8 within the myeloid cell, and initiates an entirely new immune response by that patient for their own tumor.
So I've seen some use of combining T cells and NK cells to activate both the adaptive and innate immune systems against cancer. Why the focus on myeloid biology? What's the case for doing that?
Well, in this case, the myeloid cells are really where the earliest part of the innate immune system recognizes these foreign antigens. And by stimulating them, the innate immune cells can do really three things. First and foremost, they will what we call eat that foreign body and destroy it. Second, those myeloid cells elaborate certain chemicals that we call chemokines and cytokines and attract new, fresh elements of the immune system into the tumor to help destroy it. And then thirdly, as I mentioned earlier, once they destroy the invader, they take the pieces of that and present that on the surface of the cells to train the adaptive part of the immune system to further recognize and continue to destroy other elements of the tumor.
Bolt's lead experimental therapy is BDC 1001. That's in development for HER2-positive colorectal, endometrial, gastroesophageal, and metastatic breast cancer. Why start with HER2-positive cancers?
That's a great question. And what we wanted to do at Bolt was really focus in on some, I'll call it fewer things that were novel about our initial treatment. So initially what we decided to do was choose a common therapeutic, which we call trastuzumab, which is the generic name for a drug called Herceptin. And trastuzumab has been used widely to treat tumors that express a tumor antigen that we call HER2 and inactivate that. But by using a known tumor-targeted agent, we understand what its safety profile, what its specificity is. And what we're actually doing then is just testing what this novel linker payload does without adding additional levels of complexity to this. HER2 positive cancers are ones that are still killing patients, and one where there's obviously an important unmet need that we felt we could address.
Trastuzumab is a therapy in its own right. Do you get any therapeutic benefit from using that as your targeting agent, or is it just a targeting mechanism for you?
Well, it's a great question. And the trastuzumab that we use to target the tumors in this case, it really just is a geolocator. It retains its activity as a targeting agent against HER2, but most of the patients who are coming into our study have already been pretreated with other HER2 targeting agents and have failed. So they're not responsive to these. So again, all we're relying on the trastuzumab to do is target our agent to that tumor as a geolocating system.
So if they're refractory to the trastuzumab, does that affect its ability to target those cells in any way?
No, because they still express HER2, believe it or not. That HER2 usually has mutated in some way so that it becomes unresponsive. But the antibody is still able to bind to HER2 and bring our immune stimulating payload into the tumor environment.
Well, what's known about the safety and efficacy of BDC 1001 from studies you've done to date?
Well, so we've just completed what we call a dose escalation phase 1 portion of our studies. And this is what we do in oncology to initially bring a cancer therapeutic to patients. We start with relatively low doses that we believe will be non-toxic, and then we dose escalate to higher and higher doses to understand the safety profile, but also understand where how much drug it takes to get the efficacy at the end of the day. And in this case, we were able to dose all the way through our dose escalation and show that trastuzumab, in this case, it's what we call BDC 1001, has a relatively safe profile and is well tolerated by patients. Gives us a lot of flexibility both in how we dose, but what we can combine this agent with to actually get a better efficacy along the way.
And is the phase 1 study in HER2 positive cancers across the board or specific ones?
This was HER2 positive cancers across the board. So we had, in this case, 16 different tumor types from patients that came into the study. And we were able to show, again, both a well-tolerated safety profile, which was unusual in this space. Part of the history of the immune stimulator space in oncology has been high toxicity, low tolerance to these, and part of the magic of what we're doing at Bolt, by the way we build our molecules, is to deliver these very potent immune agonists and yet have a very safe profile along with that.
As you think about the development path forward, would you look at the phase 1 results to narrow the indications, or would you expect to make it HER2 positive across the board again?
Well, obviously, at the end of the day, we'd love to have a broader indication to be able to impact, all tumor types that have high expression of HER2. In this case, for a small company, our success is going to be based on focus. And so we've chosen a small number of those 16 different tumor types that we think we've got good evidence and some rational reasons for why we should go forward and focus on those. And having, hopefully, with some positive outcomes of those, we'll have the opportunity to, continue to both fund the company, but as well build on those successes, maybe for some early approvals. And then we can come back and then broaden the tumor types through additional clinical studies.
Last year, you had discontinued development of another therapy, BDC 2034, because of off-target toxicity. What did you learn about ISACs from that experience, and how has it shaped the work you're doing going forward?
Well, that's a great question. Importantly, there's two parts of the drugs that we have. One is that targeting antibody that is designed to very specifically get things into the tumor microenvironment and onto the tumor cells itself. And then the other is the payload that stimulates the immune system. In the case of 2034, it turned out that the the antibody that was our targeting antibody not only recognized the antigen on the surface of tumors as we desired, but it also cross-reacted to another family member in that family, which was on granulocytes of patients. And it led to what we felt was an unacceptable toxicology profile that we didn't want to take into humans. So we terminated the project. It's still a project of interest to us. And today we're working on I'll call it a version 2.0, where we have antibodies that are much more specific than that initial one. And we'll come back and advance that toward the clinic as time goes on here.
Bolt is a public company. It's been successful at raising significant amounts of capital, but its stock is down sharply from its highs. It's certainly not alone in that case with other biotech stocks down, but it's got a $56 million market cap today and is trading around $1.50. What's the discussion with investors like today?
The space that we're in, which is the immune stimulating space, has had some complexities over the last couple of years. Again, as we talked of getting, a safe profile into patients that can actually eliminate tumors, and other companies ran into some speed bumps along the way where their toxicities precluded them from moving things forward. I think the recent data that we have at Bolt says that the way we are making our molecules can actually deliver on the promise that these immune stimulating compounds have. And as such, we're actually seeing investors begin to take notice again and watch the space much more closely. So what we're trying to do as a team at Bolt is focus on execution, delivering clinical results for our patients. And we believe that the investors will follow once we're able to do that.
And how far will existing cash take you, and what's the thinking on raising additional capital from here?
Well, today, the cash we've guided, the cash takes us through 2025. So we've got more than 2 years of cash available to us, allows us to take the BDC 1001 program through this next set of phase 2 studies, where we're able to look at, as you mentioned earlier, a smaller subset of tumor types to really show what this agent can do. And in addition, it allows us to take, earlier elements or other drugs in our pipeline, bring them forward into the clinic as well. And so it'll give us the opportunity to have some, what we think are important catalysts to increase investor interest, to hopefully increase the stock price as well. And with those successes, we'll have some discussions about the best timing for raising funds in the future.
Randy Schatzman, CEO of Bolt Biotherapeutics. Randy, thanks so much for your time today.
Great to be with you again, Danny. I appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.